Hi, everyone. I'm Abriana. I'm part of the STEM Starters Board, and thank you so much for being here. We have a grad student, Aaron, who's going to talk to us about his research today. So we're super lucky. Aaron, can I ask, what got you into science? What led you here the most?
Well, I got into science. It's kind of a long story, but I mean, I'll try to make it short. So, I, going into college, did not really consider science as a career for me. It was just something like I knew that scientists existed, but I never really knew the steps that I needed to take to become one. And I honestly, like up until college, never really had an interest in becoming one either. Um, in high school and even early in college, I was really interested in language. So I thought that I wanted to do linguistics when I was early on in college. And it just so happened that the university that I was at did not have a linguistics program. So I ended up in a psychology program instead because I liked psychology. And I pictured myself doing kind of like a almost like a clinical psychology kind of route with specifically going for like wanting to work with either kids with divorced parents or being like a marriage counselor kind of thing. And maybe doing some like clinical psych related stuff, some clinical psych related to that. And I realized actually that when I was in my psychology program, then of course they also didn't have a clinical focus. It was all experimental psychology, and I didn't know that going into the program. So I actually switched out of the psychology program and into a separate program that was more on counseling and therapy type stuff. And I was in there for a year. And some of the classes that we were taking at that point were I had to take some classes on like child development and human development and that sort of stuff. And I remember at the time there were other things going on in my life too that kind of just made me think more about the brain in particular and the role that it plays. And so this other stuff that was going on in my life, like there were, I was going through like some stressful situations, and at the same time I was learning in some of my child and human development classes like how critical certain periods of child and human development are, and the fact that excessive stress during certain phases of child development can result in bad long-term outcomes. So like you don't want your kid to be too stressed out when it's a baby, because then even in adulthood, those effects can be long lasting, and the child might have some predispositions for either mental disorders or other health conditions and stuff like that. So I again kind of realized through that the brain was really kind of driving a lot of that. So it's like the brain is super susceptible to stress, and when the brain is not well balanced, and when there's this dysregulation of stuff that affects the brain, then it can have really bad long-term outcomes. So then I realized that I could also, like, the reason I went into the therapy and counseling kind of program was because I wanted to help people. But then at the same time, like realizing in taking these classes, I was realizing I could help people a different way through science, and not necessarily sitting down face to face with an actual patient or client or something. I realized that, we can actually have impact as researchers and kind of inform the field of, physicians or psychiatrists or counselors and stuff, and they can use our research that we're doing to help them make decisions about how to treat their patients. So that's kind of what helped me realize that I wanted to be a scientist and not just not a counselor or a therapist or clinician.
That's wonderful. It comes from such a good place. Thanks. So now that you are doing science regularly and you're working in a lab, what's the best part about it for you, and what's something that maybe you struggle with the most?
So what I like the most, I think, about science is the creativity of it. I like the fact that, I can go into an experiment and kind of have control over what I'm designing, because I get to kind of pick to an extent what the research question is. Obviously, you're kind of like picking a lab based on general stuff that you're interested in, and then, the dynamics of certain things that you're working on in the lab are kind of a balance of what you want to do and what other people in the lab are doing. But you do still have control over, you have say over what experiments you're going to run and what specific question you want to tackle. And I really like that ability to design experiments and kind of like think things through. And then if something goes wrong, like I'm in charge of troubleshooting my own experiments. So I like the responsibility aspect of it, and the creative control.
Mm-hmm.
In terms of stuff that I struggle with, I guess like, a lot of times things don't always go well with the experiments. And sometimes you're really banking on getting some effect that you think is going to happen, and then you don't get it. And then you have to kind of go back to the drawing board. So, sometimes going through that repeatedly can be like a little bit demoralizing, or make you like question, certain things about, how you're thinking about a problem. But, it's important to just like stay cool and, realize that that's part of how science works. Like, and that's always going to be the case. There are always going to be those kinds of issues that pop up. But it's important to just like keep the big picture in mind and like stay focused on that. And then you can pretty much get through anything.
Absolutely. Thank you. All right, well, I'll let you go ahead and start. Everyone, if you have any questions, feel free to pop in, unmute yourself and ask them, or put them in the chat and I can do the interrupting for you. And we highly encourage you to ask questions. All right, I'll go ahead and mute myself and let you go ahead. Thanks.
Okay, yeah, thank you. So the title of my talk is Past, Present, and Future, How Our Memories Shape Our Behavior. And like Aubriana said, please ask questions whenever if something pops up. I'm happy to answer anything that you guys have. So this talk is going to be about how our brain, which, is kind of the basis of everything that we do and who we are, how our brain controls our behavior through our past experiences. So things that have happened to us in the past have impact on who we are and will shape the decisions that we make and the actions we take in the future. So I open up with this picture, comes from Salvador Dali, who was a surrealist painter. He painted this in 1931. It's called The Persistence of Memory. And it's a pretty, weird picture. You can notice some clocks are looking like they're melting. This to me is an interesting, I really like the choice of the title on top of what the painting actually is, because I feel like the clock melting is a way of, time kind of changing, melting, affecting stuff that's going on. And we have like, well, there's the persistence component of time too. Like time is always going to be there, and over time, things change. So over time, we'll see later on, memories change too. And there is a persistence of memory, but we'll also see later on in this talk that our memories are actually reconstructed, and they're malleable, so they will change over time. A memory is not a perfect picture of what happened in the past. A memory is kind of our redrawing of a past experience based on some things that we might remember. And our memories, depending on what other things are going on, might change. So we're going to see later on how some of that stuff happens. But I just kind of wanted to open with this because I think it's an interesting piece. So why should we study memory? Well, why should we care at all about anything that's going on with memory? Well, first of all, I would hope that people just think memory and the study of it is just cool in and of itself, but we also study memory because we want to help people and treat certain disorders. And so 2 common disorders that are associated with memory are post-traumatic stress disorder, or commonly known as PTSD, and Alzheimer's disease. So these are just 2 of many different types of memory disorders. But when you think of things that are related to memory, these are 2 of the most common disorders that pop up. So they're interesting because they both reflect different aspects of memory. So in post-traumatic stress disorder, having a memory is painful, but in Alzheimer's disease, not having the memory or losing a memory is painful. And it's painful for, in the case of PTSD, it's painful for the patient, it's painful for their families because they're remembering an experience that has happened to them in the past, which was a source of trauma. And, it's tough for people to get over traumatic situations. So a lot of times, they need special help and treatment to work their way through that. And in Alzheimer's disease, this is also painful for the patient and the family as well, because, the patient is losing these valuable memories of times that they shared with their loved ones, and then the family then is feeling like they're losing someone that's close to them because they feel like it's no longer the same person sometimes. So studying memory allows us to kind of address certain problems with these diseases, and we hope to one day develop treatments for these sorts of things, but it's they're very complex, and it's taken decades of work so far, and it will take probably decades more before we start getting to a point where these disorders and diseases are completely treatable or preventable. And so a few things that I'd like to go over in this talk are these 3 points. So first one is, how are learning and memory defined and studied? So like, how do we define and study learning and memory in the lab? And then how are memories created and stored in the brain? And number 3, how do memories go bad and how can we fix them? So let's start with, what are learning and memory? Well, we define learning as a lasting change in behavior as a result of experience. So when something happens to you, there might be a bunch of different things that you notice that don't have an impact on you. Like right now I'm looking out of my window and I see some sort of bird feeder, and, apart from talking about it right now, it's not going to have an impact on me as a person or anything. Like, it's not going to affect my behavior or anything. But, there are other things that can happen as a result of experience. So for example, if I get bitten by some sort of animal or something, like I'm going to learn to avoid that animal in the future. And that is a result of the pain that animal inflicted on me, and I'm changing my behavior to avoid that animal. So that is a learning process. So that's why we define learning as a lasting change in behavior as a result of experience. And it's important to realize that the behavior is really the only thing that we can measure that indicates learning without looking directly at the brain. So, you know, in normal everyday life, we're looking at behavior. And so memory, learning and memory are also broken down into two broad categories. So we have declarative memories and procedural memories. So declarative memories can then also be broken down. So there are semantic memories, and these are things like who you are. And then there are episodic memories, which are things that have happened. And so like in this little figure here, you know, a declarative memory would be, you know, the ability to remember certain things like maybe your own phone number, friends' phone numbers, stuff like that. I know nowadays might not be so relevant because we kind of just like, you know, put our contact list in our phone. But back in the day, we used to have to actually remember our friends' phone numbers. I can still actually kind of remember some of my childhood friends' phone numbers from like, you know, 6th grade and stuff. We used to have to memorize all that stuff before we could put it in a phone. You know, memorizing your address and friends' addresses, where you parked your car, what even a parking sign means. These are all types of declarative memories. And then there are procedural memories too. And so these are basically just motor memories. So, you know, when you're first learning some sort of physical task, you're probably very clumsy at it and you don't really, you know, perform well. But the more you do it, the better you get at it. So you're refining your muscle control and your brain's ability to, you know, exert control over your muscles and kind of see, you know, figure out optimal ways to achieve some sort of goal. And these kinds of things are all broken down into, or all encompassed in procedural memory. So how do we test learning? I kind of hinted at it with the definition of learning, but let's think of an example here where you want to get, say you want to get a car or a driver's license. There are 2 different tests that you have to go through to show that you're going to be allowed to have a car. So one of them is the written test. The other one is a driving test, a performance-based test. And actually, I mean, both of these in a way are performance-based. Here you're just performing on the written component of the test. And here you're performing on a physical part of the test, which is, you know, the real life application of what you've learned. So it's pretty clear here that learning is being tested by the behavioral performance. So we're looking at the behavior of what a person is doing, and that is how we're defining whether or not that person has learned. Any questions on anything that I've talked about so far? Okay. So now in the lab, you know, things are a little bit different when we study learning and memory and stuff in the terms of, you know, we're not always working with humans. You know, there's we can do a lot of stuff with humans, but at the end of the day, if we want to figure out what exactly are a lot of the neurobiological components of memory, if we want to develop, you know, certain types of treatments and stuff, it's important that we first do some stuff in other organisms besides humans so that we can confirm that these sorts of treatments and therapies are safe to use in humans. So we use, you know, different what are called model organisms to do this sort of stuff. And so a lot of the stuff that I'm going to be talking about today uses a mouse. And so mice are mammals just like us. Their brain is a lot smaller than ours, but it has a lot of the same structures in it. And so that's why we like to use the mouse. And to study memory in a mouse or a rat, basically just any type of rodent, we use different variants of this box, which is called an operant box. And depending on what you want to study in terms of learning and memory, there are different things that this box can do. So for example, you can put a mouse or rat in here and have them press a lever to get a reward, which comes from this little dispenser here. And so there will be food in here, and then it'll get sput out once the mouse presses the lever, and it'll come here, and the mouse can go and pick it up and eat it, and then press the lever again and get another one. And so you can kind of learn, or you can see how the mouse learns the ability to press the lever, and you can combine that with different lights or cues to signal when a reward is available or not. You can also use an electrified floor of the box and administer small shocks to the animal to test its ability to learn negative memories. The ability to press a lever and get a reward is generally what's called a reinforcing thing. So the mouse or rat is like motivated to do that more because it's getting something that it likes. But then you can also administer a small shock, and then it'll do certain things to try to avoid that shock. And just to be clear about everything, when we do animal research, we do everything very carefully, and we follow what are called the 3 R's of animal research, which are replace, refine, and reduce. So we are trying to replace animal research altogether when we can. So for example, if there was some sort of analysis that we could do that did not require us to use mice or rats at all, then we would do that. We would not use lower organisms or any animal model for something if we don't have to. If we can, just do things on a computer or do things in humans or something, then we would prefer to do that. Refine means to refine the experiment so that the harm to the animal is minimized. So we're always trying to make sure that, whatever, if we're doing anything that's uncomforting or uncomfortable for the animal, then we try to minimize that as best as we can. To an extent, we would have to do some aversive things, but we're always trying to make sure that it's the least amount of harm that we can possibly do for the organism. And then also reduce. So we want to make sure that we're using the fewest numbers of mice and rats possible for these types of experiments. So, and all this stuff goes through a very heavily vetted process where we have veterinarians and members of the public that guide our experiments, are what are called animal protocols. So, if there's anything that's over the top, they say, hey, you can't do this, or you need to change some stuff. So I just want to be clear about that before I start talking about these, some of these experiments. So I kind of hinted at some of this stuff, but now we can kind of go through. I have a list of different things, and we can kind of guess which organisms can learn. And remember, we defined learning earlier as a relatively permanent change in behavior as a result of experience. So let's start with humans. I think we can all agree that humans can learn. Dogs can learn. Yes, we know that, you can teach your dogs tricks. Cats learn where the litter bed is. Mice learn to press levers and stuff. Birds learn where their food stashes are. Fish learn to, revisit certain areas of the ocean or lake. Salmon travel thousands of miles to go back to the same spawning site that they were born in. Even insects can actually learn. So you can do, some of the same stuff that I was talking about with either rewarding or aversive conditioning, where you can put an odor cue with a small shock for a little fruit fly, and it'll learn to avoid the area that smells like where it got shocked, because it knows that if it smells that, it'll get shocked. So that's learning. There's actually even evidence that some plants can learn. They don't even have a nervous system. And ciliates are single-celled organisms, and there's evidence that they can also learn. And these particular plants and ciliates, the types of learning that is going on in these organisms is not fast. It's on extremely slow timescales, to the point where, you know, if you were just looking at a plant, you wouldn't guess that it's learning. You would have to, you know, like monitor it for several days on end. And same thing with the ciliates. Like it takes a long time, but they do learn. And then rocks are not organisms, and unsurprisingly, they cannot learn. But, you know, almost anything in the biological kingdom can learn. You know, in terms of mammals or vertebrates in particular, like we have more developed nervous systems, and that helps us learn things very quickly. So that's what sets us apart from other things like, you know, plants and ciliates and insects. And yeah, humans, you know, there's a lot of debate about the things that help us learn very quickly. I think Sergio's talk at one point talked about things that make human brains unique, and there was you know, stuff with axonal branching. But yeah, so whenever someone tells you that bugs can't learn or fish can't learn, stuff like that, like they can.
Can I pop in with
so like I said, sorry.
Oh, I was wondering, could I pop in with a question? Does
Yeah.
the size of the brain directly correlate to like how many memories? And I mean, maybe we have no idea, but like to how many memories an animal can store?
No, it actually, we don't think that it does, but yeah, it's a difficult thing to test. And we don't think that it does, for example, because birds actually do not even have a hippocampus, which I haven't talked about yet, but we'll get to in a few slides. Humans have a large hippocampus, which the hippocampus is an important region for learning and memory in mammals. But birds don't even have one, and they can remember thousands of different locations where they've hidden their food and stuff like that. And you know, I think dolphins and whales, well, whales in particular, I think, you know, some of them have pretty big brains, and they're not necessarily better at remembering things as far as we can tell. But you know, it is hard to test, you know, how well they know certain things just because they're so vastly different from us. Right. But we don't think that the size of the brain directly correlates with memory storage. And yeah, so in mammals, the brain is an important structure in guiding how experience gets translated into behavior. So your brain, first of all, is taking sensory input from the outside world, and it's directing attention to certain things that, you know, it thinks are important. So based on the stuff that's going on around you, you might find something more important than others. And you'll pay attention to those things. And when you pay attention to some things, you'll learn associations about the outside world. So an association I'm defining as like a pairing of two objects. So, or two events actually. So for example, like when you maybe like, yeah, like when you hear a bell at school, then you know you're either like late to class or class just ended or something like that. So that's learning to pair the bell with an event of like, let's leave class or let's hurry up and get to class or something like that. Or, you know, like a car horn, you know, like if you just hear a car horn, you're going to know like, okay, someone's honking at me or someone's honking at someone else. But you know that you've paired the car horn with, you know, someone being frustrated or something like that behind the wheel. And then you do that because you've had past experience with that. So the first time you hear a car horn, you might not, associate it with anything, but if you, see someone getting mad at the car, or you see someone in the car getting mad at someone else as they're like honking on the horn and stuff, then you can, kind of remember like, okay, the car horn means that somebody's mad at somebody else or something like that. And then based on that, you can control your movement. So, if you've had bad experiences with car horns, like if you've been in an accident and you heard a car horn right before someone hit you, or right before you hit someone else, then, you're gonna, next time you hear a car horn, you might like tense up because it was a bad experience for you in the past, and you have this like strong reaction to it. So that's the brain is doing all of these things. It's regulating what you see in the outside world, or what you pay attention to in the outside world, and accessing past memories, and then controlling your future directions and future movement through all of that. So now we're going to home in on a region called the hippocampus a little bit. So this particular case study was an important one in pointing scientists and physicians to a certain area of the brain called the hippocampus. And so this came from, this knowledge came from a patient. So that's who this guy is. His name was Henry Muleyson. And at the time he was only known as HM. They couldn't release his full name for patient confidentiality, but after he passed away, then they, identified him, and now his brain has been dedicated to science, and I forget what university it was at, but yeah, people are studying it. But he had temporal lobe epilepsy when he was younger, and a common treatment at the time for that was what's called a lobectomy. And so a lobectomy was when basically doctors would just go in and they would make an incision in the skull and extract part of the brain. So this epilepsy causes seizures, and temporal lobe epilepsy in particular affects this area called the hippocampus, or the hippocampus is inside the temporal lobe. And so when he underwent this lobectomy, and he was only 26, I think, when he went through it, he lost the ability to form new memories after the surgery. So the doctors took out his hippocampus as part of the temporal lobe lobectomy. And after that procedure, he could no longer form new memories, new declarative memories, I should say. He could no longer, I think he could not remember his doctor's name and stuff like that. Like it took him, he was alive for decades and was, every day, like not remembering anything that had happened after his surgery, but he could remember everything from before his surgery. So he still had all his, childhood memories and stuff. And he could form, if I recall correctly, he could form procedural memories. So he could learn new motor skills and stuff, but he could not remember, details about the world around him after that lobectomy. So this was one of the case studies that pointed doctors and scientists towards the hippocampus as like this important structure for learning and memory. And so now we're going to dive in a little bit more to the brain. So your brain is about 2.5 to 3 pounds, makes up 2% of your total body weight. It has 2 hemispheres. So this top picture is a view from the top. And so you have a left hemisphere and a right hemisphere. All of it is encapsulated by the skull, and it gets its nutrients through blood and cerebrospinal fluid. There are about 86 billion neurons in this brain, and neurons are the primary type of brain cell. Then there are another 80 billion non-neuronal cells. So there's a lot of cells here in human brain.
Aaron, can I ask
so these
question?
neurons, yeah.
I think this might be kind of silly, but does everyone's brain have the same like ridges and indents that like we're looking at here, or is it kind of unique to everyone?
For
like
the
I know
most
everyone
part,
has the same areas, but
yeah. So these, these gooey looking parts on the outside are called gyri and sulci. So a singular one would be a gyrus and a sulcus. And yeah, they are pretty typical of every person. There are, certain disorders and stuff where some of them might be like larger or smaller, but overall, they're pretty much the same. And there was a neurologist, I think his last name is Brodmann, who went through and kind of divided all of these up into what are called Brodmann's areas. And they were super influential throughout like the second half of the 20th century. I don't know how commonly they're used now, but they were definitely very important earlier on. And I think they might mainly be used now by like more of neurosurgeons and stuff. But I think overall, most of the human research has kind of just shifted into more defined, like, how do I say it? I guess less defined areas that are more kind of like function specific, because it seems like some of his areas were just kind of like arbitrarily picked out or whatever. But yeah, to answer your question, yes, they're generally the same throughout most humans.
cool. Thank you.
So neurons are about 20 micrometers at the soma. So that's very thin. That's about a quarter of the width of human hair. There are about 86 billion neurons in your brain and about 125 trillion synapses. And a synapse, as we'll see, is the area where neurons connect with each other. And so each neuron connects with a bunch of other neurons and connects with so many neurons that you end up with 125 trillion synapses. And to put this number in perspective, if a synapse was the size of a grain of salt, you would be able to fill about 125 classrooms with it. So you could fit about 1 trillion grains of salt in a classroom. So if you have 125 trillion synapses, that's 125 classrooms. That is a lot of salt. And of course, each synapse is incredibly small. The synapse is on the order of 20 to 40 nanometers. So that's why we're able to fit so many of them in our brain. Yeah, neurons are the primary cells of the nervous system. And like all cells, they contain organelles. So here are just a few of the organelles. I didn't put all of them on this diagram. Didn't want to make it too overwhelming, but yeah, they have, the important stuff: nucleus, mitochondria, Golgi apparatus. And then there are 4 main components of a neuron, and this is kind of what sets them apart from other cells of the body. It's kind of the morphology and the things that neurons can do. So they have these things called dendrites, and these are basically going to receive input from other neurons, and then this input then gets passed down. So if you're looking at this neuron, the information flow is going to go from left to right down this way. And so information gets absorbed in through the dendrites, signals get absorbed through the dendrites, and then it gets passed to the cell body, which is also known as the soma. And then that gets sent down it's called the axon, which is this long stretch here. And then that gets, then that eventually reaches what's called the terminal, which is the end of the neuron. And then it's converted into a chemical signal. So all the signal that's going on inside of the neuron is an electrical signal. So there's a lot of different ions flowing around, and you can measure these using electrical equipment. And then when it reaches the terminal, it gets converted into what's called a neurotransmitter, and it gets passed on to the next neuron. So we'll explore that a little bit more here. So here's a diagram I have of two neurons. So this neuron on the bottom is called a presynaptic neuron. And this neuron here is called a postsynaptic neuron. So that means the information is going to flow from this neuron to this neuron. So if we explore this synapse in more detail, the synapse is this area in between the two neurons. And it's a very small area. Like I said, it's about 20 to 40 nanometers. And then you have what are called receptors that sit inside this synapse. And so the receptors are on the postsynaptic neurons. So this is again, the presynaptic and the postsynaptic. And so you have neurotransmitters that are inside the presynaptic neuron. And remember I said there's an electrical signal that goes through the neuron. So when that electrical signal reaches here, it pushes the neurotransmitters out into the synapse, and then they bind to the receptor, and that allows other ions to flow into this postsynaptic neuron. And then this postsynaptic neuron carries out this electrical signal itself, which is called an action potential. Any questions on this? Okay, and then the neurotransmitters end up like, the neurotransmitters do not usually enter the postsynaptic cell. Usually they fall off the receptor and either get like degraded in this synapse, or they get uptaken back into the presynaptic cell and like recycled, basically. And so one thing I forgot to mention is that when, so I said after the neurotransmitter binds to this receptor, then there's this influx of ions that comes into this postsynaptic neuron. And when that happens, one ionic species that's uptaken is calcium. And so calcium then affects other what are called signaling molecules, which travel, they make this long voyage all the way from the synapse to the nucleus of this neuron, and they start interacting with the DNA in the nucleus. And so when that happens, a protein is produced. So we can use this, it's like a genetic or a protein signature of the cell. So when this happens, say we have some active neurons here, then shortly after they're active, there's this what's called upregulation of this protein in the nucleus. So we can use the fact that this protein is upregulated, and it's almost like seeing a footprint of neurons that were recently active. So now we can look at these neurons in a microscope and see which ones were recently active when we, there's a technique called immunohistochemistry that allows us to label that protein in the nucleus, and we can see it and look at it in a microscope. So we can see in the brain which neurons were recently activated. And so that's what this looks like. So this, these red dots are what's called CFOS, which is the protein that's upregulated. And so you can use this protein to see which neurons were recently activated. And this particular area that we're looking at here is a region called the dentate gyrus of the hippocampus. So this is one area. In this particular image, we're looking at mouse hippocampus. And so that means that all of these red cells were recently activated as this mouse was, performing some behavior. So we can use this. Now we know, remember that the hippocampus is important for learning and memory. And now we can see the physical basis of which cells were recently active. So now we can start to ask the question of which cells contribute to memory formation, or how do cells in the hippocampus contribute to memory formation. So I'm gonna go over this experiment with you guys. It's a really cool experiment because what the researchers did is they kind of tricked a mouse into thinking that it had an experience when it didn't. And it's causing the mouse to become afraid of something that it should not have any reason to be afraid of. So when this experiment was published, they nicknamed it the Inception experiment. I don't know if you guys have seen the movie Inception, but it's a movie with Leonardo DiCaprio, and the premise is that they are trying to implant a false memory into somebody. They're trying to get somebody to do something, so they're trying to trick them into thinking that something happened in the past that didn't actually happen. And so researchers actually were able to do something quite similar in a mouse. So what they did was they have a context, and they put the mouse in this context. It was in one of the conditioning boxes, or the operant boxes, like I showed you before. So it has an electric floor, and so there's a shock that comes out of it. And the mouse, of course, doesn't like the shock. So it's afraid of that, and it's afraid of this context then, because it knows that there is a shock in this context. And so when this happens, there's a set of neurons that are activated as the mouse is doing this. So that would be the neurons, the yellow neurons are the ones that were active in context A, and then these like grayish ones were just not active. So then they had a way of tagging those active neurons. So they used the CFOS method, like I showed you before, to label neurons based on their CFOS activity. So now they can use that footprint and say, okay, let's target these neurons that were recently activated, and then they're going to reactivate them in a new context. So they put the mouse in this new context, which is context B. And when if the mouse was normally in this context without, any manipulation, there would be a set of cells that would become activated as the mouse is in context B, and they would be a little bit different from the ones that were in context A. And so the mouse would have no reason normally to be afraid of context B. But what these researchers did was they reactivated the ones from context A. So they took, they used a manipulation to reactivate these neurons as the mouse was in context B. So that's what this would look like. And then something interesting happened, because the mouse became afraid of context B, but there is no shock ever given in context B. And so the next day, they wanted to test whether this was a long-lasting manipulation. So at this point, this isn't really so much as learning a new memory, because remember we said learning needs to be a lasting change in behavior. So at this point, the mouse was remembering something from context A, but we don't know if it's formed an association with context B yet. So what they did on the third day was they put the mouse back in context B without doing any manipulation, and the mouse was still afraid of the context. So that means that the researchers were able to reactivate this set of neurons that were associated with a bad experience. When they reactivated them in a new context, the mouse became afraid of that context without ever experiencing a shock in that context. And so now a big, yeah, sorry, go ahead.
That's so interesting. It makes me wonder, like for someone who might have PTSD, like if they have like a surge of like memory, or like a traumatic response in a new place, could that new place also become like a source of stress for them. Is that
Yeah,
like
that's exactly basically where I was going to go with this. Yeah, so that's the big open question right now. Is what we have seen is the mouse is afraid when it's in context B. Yes, but we don't know then if this fear here is also due to the same patterns of reactivation. And if we were able to see that, then it could point towards a potential treatment for PTSD based on, targeting these neurons that were active during some experience. So yeah, that's one of the big open questions right now. A lot of people are looking at how we can use these, use this type of knowledge about how neurons reactivate to try to develop better treatments for PTSD. And so one way that we're starting to get at this question here is by using a technique that's called calcium imaging. And so in calcium imaging, we take a really tiny microscopic lens, and it's so small that we can put it inside the brain of the mouse, and we can attach it to the mouse's head, and the mouse can run around and perform behaviors. And here you can see cells are active as the mouse is moving around. I'm not showing a behavioral video of the mouse here, but this mouse is moving around, and these cells are firing as the mouse is moving around. So we can use this technique to see which cells are active during an experience. So it's even, in some ways, it's better than the CFOS method, because now we can see specifically within any type of behavior which cells are active during different parts of the behavior. So for example, like if the mouse is, so like on this slide, for example, like when we are looking at, when we're defining fear in the mouse, it's defined as freezing behavior. So the mouse is like freezing in anticipation of the shock. And when it's not afraid, it's like moving around a lot more because it's less afraid. And, so one question that calcium imaging could let us do is, we could look at specifically those moments when the mouse is freezing to see if those neurons are reactivating. So then we can draw even a more direct link between the neuronal activity and the behavior. So that's like, the next big question of the field. Okay, so I think that between these two slides now, we've seen that these memories can be changed experimentally. So yeah, we can target those neurons using genetic strategies to manipulate memory, but it turns out that you don't even have to do that in humans. Episodic memories are already extremely malleable. So based on the work of this scientist, Elizabeth Loftus, she found a lot of different ways that you can manipulate episodic memories and kind of trick people into thinking that certain things happened that didn't actually happen. And her work was so important that she became an expert on eyewitness testimony in court cases. So based on, because of her work, there is now a lot of regulation about how lawyers can get eyewitness testimony out of people. So based on the questions that lawyers or police ask people, sometimes those questions, the way that they phrase those questions can affect the answers that people give. So for example, one of the common experiments that, one of the most impactful experiments that she did involved showing participants a video of a car passing. And then she would ask 3 different variants of this kind of question to different participants. So one question that she would ask one set of participants was how fast was the car moving? Then to another set, she asked how slow was the car moving? And then to a third set, she would ask like a neutral question, at what speed was the car moving? So she found that if you ask people how fast the car was moving, they'll give you a much higher answer on average than either of these other two groups. And then likewise for how slow was the car moving. So if you ask a group how slow the car was moving, they're going to give you an answer that's much slower. And basically it's important to phrase things in this kind of neutral language, like at what speed was the car moving? So just to give a more concrete example, so like let's say this car was actually moving at 45 miles per hour. And if you're an eyewitness and you saw this, an accident happen with this car moving and it was at 45 miles per hour and you saw someone get hit, and a lawyer or a cop asks you, how fast was that car moving? Like it was moving pretty fast, wasn't it, for that person to get hit like that or for that car to get hit? You'll give an answer that's more on the order of say like 60 miles per hour or something, even though the car was really moving at 45. And then if they asked you how slow was the car moving, you'd give a lower answer. So I don't know, maybe like 30 miles per hour or something. But then if the question is neutral, at what speed was the car moving, then you'll give a more reliable answer. And so, yeah, her work, this is just one example of all the stuff that she did to show that episodic memories are malleable, and that based on different factors that are going on in the ways that someone processes a memory after the fact, there are a lot of different ways that memories can be manipulated without us even realizing it. So our memories are kind of being reconstructed from like this past information that we have, but in no way is our memory of something a perfect picture of what happened in the past. And so another example of this, going back to mouse research, is that experiences within short timescales become linked in memory. So there's a really cool experiment where some researchers put a mouse in our context A, and they gave the mouse a shock. And of course, the mouse became afraid of the shock. And importantly, there is a scent that's associated with context A. So when the mouse is in context A, there is a scent. Let's just call it vanilla. So they put like this vanilla scent in context A. So the mouse has like this spatial, like it can see everything that's in context A, but then it also smells vanilla. And so then they put the mouse in a new context called context B. And the important thing here is that there's this short time scale that's important for this. So it has to be 5 hours or less for this to happen. So what happens if you put the mouse in a new context B, in 5 hours or less, and if you put an object in context B that smells like context A, so an object that smells like vanilla, the mouse actually becomes afraid of that object and will stay away from it. And then the next day you can test it in a new context. Whoops, and the mouse will still be afraid. I forgot to put the little exclamation points here, but the mouse will still be afraid of this object. But if this time window is over 5 hours, the mouse will not be afraid of the object in context B, and it will not be afraid of the object in context C. So this again points towards the fact that there's a lot of different molecular signaling that's going on that controls how memories are formed and maintained over time. And then, of course, there are a lot of other factors that influence how we learn. So kind of going back to Elizabeth Loftus's work on eyewitness testimony, she showed that there's a lot of effects of rehearsal. So how we replay the memories, or how often we think about the memories after the fact, that can affect our recollection of a past event. There's also what's called the valence, which is whether something is good or bad, or whether, like if we get a reward, if we get like a cookie for doing a good job, or if we get a shock for doing a bad job. The intensity of the outcome. So, whether we get like 1 cookie versus 5 cookies, or like, a small shock versus a big shock. And then the co-occurrence. So if there are a bunch of different, stimuli happening at the same time, it's harder for us to pay attention to certain ones. So we might not learn as reliably when there's a lot of distracting things going on. And then there's also like more health related things that affect how we learn. So there's sleep, obviously. Actually in sleep, a lot of times people have shown that when you sleep, you're actually replaying memories, and a lot of the same cells become reactivated during sleep that were originally active during some experience. And that through this mechanism, through sleeping, things become converted from short-term memory to long-term memory. So there's like this replaying of events that just happened to you when you're sleeping, and that helps convert things into long-term memories. Stress is a big thing that affects how we learn. So stress makes it difficult a lot of times to learn new things. A little bit of stress is actually good for learning, but if you're too stressed out, then it's very detrimental to learning. And then diet, the things that we eat and put in our body always affect, our performance, and they'll affect how readily we learn certain things, and then just our general health. And if we're sick with certain things, the immune system plays a big role in regulating how the brain processes new information as well, which is a pretty interesting area of research. There's, a lot of open questions in the field of immunology and how that affects learning and memory.
Aaron, we have
and so to wrap up.
Yes,
Okay.
previous slide. So Ashley is asking for the one with the different context and the vanilla smells, if there is a similarity between if there's a similarity between context A and B, and then a different similarity between context B and C, will the mouse still be scared of in context C?
If there is a
like, might it associate the another thing in the environment in context B where it was afraid?
There's a similar, okay. Oh, I see. So this is actually called, I believe it's called transitive association. So yes, there are that has been shown. It hasn't been shown in this direct set of experiments, but yeah. So if you have, say, like a let me think. If you have a like a bell that predicts a foot shock, and then later on you start conditioning something like a different sound before the bell, then depending on, it kind of depends on the parameters of the experiment, but there are ways to do that. So then the new sound then will start to predict the shock itself.
Awesome, thank you.
Was that kind of what, that's my interpretation of what the question was asking. Is that a correct, satisfactory answer? Okay, so yeah, and so now, the next step is to use this knowledge to treat memory disorders. And so in the cases of PTSD and Alzheimer's, in some cases, people are developing, pharmacological treatments and, different medications that people can take. But there's also the potential to use just behavioral treatments or cognitive-based treatments that don't actually require you to put medications in your body. So like if this is always considered like a first round of treatment. And then if people don't respond well to these types of things, then they'll go on to medications and stuff. So exposure therapy for PTSD is when you are exposed in some way to the traumatic event that happened, and you kind of replay different parts of it with the help of a therapist or counselor to kind of help you like work through the different emotions and stuff that you're feeling at the time. And there's evidence that, so say for example, so going back to the mouse with the foot shock. So if there's a tone that comes on that predicts the foot shock, then you can put the mouse in a new context. And if you play the tone at first in this new context, of course, the mouse is afraid of the tone, but the more you play it without giving the shock, the mouse will learn that the tone no longer predicts the shock. So then it'll start moving around and doing like normal stuff. And so, that's basically the idea of exposure therapy. So you're kind of exposing someone to something that makes them fearful, but you do it over and over, and you kind of just show them that like, look, there's really nothing to be afraid of, so to speak. And in the case of exposure therapy for PTSD, now they're kind of doing like virtual reality based exposure therapies. And this isn't just for combat soldiers either. Like PTSD can happen to anybody. If you've been in any sort of accident or anything like that, like that's like those, anyone can develop PTSD basically. And like there are different types of exposure therapy for a lot of different manifestations of PTSD. So it's definitely an encouraging area of research to see that like people are starting to develop solutions that might not necessarily just jump straight to medications and that sort of stuff. And then in terms of Alzheimer's, there's music therapy. And so, it can be an effective way to actually help protect against some aspects of memory loss. And in some cases you can play, for patients with Alzheimer's, you can play a song that, reminds them of, like say their childhood or when they were, younger or something like that. And it'll actually help rescue some of the memories that people thought were lost. So it's a way of kind of like helping to dig deeper into the memories that might still be lurking around. Like it might be hard for people to actively like recollect like all of these memories that they've had, but through the help of music therapy, then you can actually like get some of those out sometimes. And so that's been a very encouraging area for, the patients themselves as well as their families, because they can see, how much happiness and joy that these patients with Alzheimer's still have. And so, yeah, to recap everything that we've covered, a lot of different organisms can learn. It's not just a human thing. It's not just a mammal thing. It can happen on very, very, very slow timescales in some organisms that you might not have even thought about. But within humans and mammals, the hippocampus is important for declarative memory. And these memories are encoded in populations of hippocampal neurons. And we can target those neurons to modulate memory. And then we are using this information to continue to develop treatments for memory disorders. And that is all that I have. Thanks, everybody. Yeah, happy to take any more questions that anyone has.
Yeah, thank you so much. This was so awesome.
Thank you.